Microbial interactions encompass various forms of symbiosis, where two or more different species associate closely. Ectosymbiosis involves one organism living on the surface of another, while endosymbiosis occurs within another organism. Endosymbiosis sees microorganisms living both inside and outside their host. Thiothrix species, a sulfur-using bacterium, attaches to surfaces and contains a parasitic bacterium. Fungi associated with plant roots often contain endosymbiotic bacteria and have bacteria living on their surfaces. Symbiotic relationships can be intermittent or permanent, and each interaction affects the larger biotic community. Microbial interactions can be positive, such as mutualism, protocooperation, and commensalism, or negative, including predation, parasitism, amensalism, and competition. These interactions illustrate the complex relationships between different microbial species. Mutualism defines a relationship where both partners benefit reciprocally. This relationship often involves some degree of obligation, meaning the partners cannot live separately and are dependent on each other for survival. Flagellated protozoa live in the gut of termites and wood roaches, digesting cellulose and metabolizing it to acetate, which termites then oxidize. Termites rely on these protozoa for cellulose digestion, highlighting a mutualistic relationship essential for their survival. Lichens represent a mutualistic association between fungi and either green algae or cyanobacteria. The fungus provides protection and minerals, while the phototrophic partner supplies organic carbon. This relationship allows both organisms to thrive in environments where they might not survive independently. Syntrophism involves the growth of one organism being dependent on or improved by another organism nearby. This mutualistic relationship, also known as cross-feeding or the satellite phenomenon, benefits both organisms involved. Protocooperation is a positive symbiosis where both organisms benefit from the relationship, but it is not obligatory. This cooperative interaction differs from mutualism as it does not require the organisms to live together permanently. Marine worms secrete mucus from glands on their backs to feed bacteria, which in turn protect the worms by providing insulation. This protocooperative relationship showcases how both organisms benefit from their interaction. Quorum sensing involves the accumulation of signaling molecules that enable a single cell to sense the density of bacteria. As the population increases, specific genes are expressed, influencing microbial community behaviors such as symbiosis, virulence, and biofilm formation. Many bacteria use quorum sensing to control gene expression, especially during infection. This mechanism helps pathogens coordinate their virulence and escape the host's immune response, ensuring successful infection. Signals in gram-negative bacteria are often acyl homoserine lactones, while gram-positive bacteria use oligopeptide signals. Commensalism is a relationship where one organism benefits while the other is neither harmed nor helped. This interaction can involve waste products or environmental modifications that make conditions more suitable for the commensal organism. In the human colon, E. coli uses up oxygen, allowing anaerobes to grow. Microbial succession during milk spoilage promotes the growth of acid-tolerant species. Initial colonizers help other microorganisms attach, and host plants release compounds used by commensals. Negative interactions among microbes include predation, parasitism, amensalism, and competition. These interactions highlight the competitive and sometimes harmful relationships that can occur within microbial communities. Predation occurs when one organism, the predator, engulfs and digests another organism, the prey. Examples include Bideo-vibrio, Vampirococcus, and Daptobacter, each showcasing unique methods of predation within microbial communities. Predation can have beneficial effects such as digestion, protection, increased fitness, and survival. The microbial loop illustrates how predation contributes to nutrient cycling and the survival of certain microbial species. Parasitism involves one organism, the parasite, deriving nutritional requirements from its host, which is harmed in the process. This relationship often involves long periods of contact and can significantly impact the host's health. In the microbial world, the distinction between parasitism and predation can blur. Depending on the equilibrium between the organisms, a stable parasitic relationship may shift to a pathogenic one, effectively becoming predation. Amensalism is a relationship where the product of one organism negatively affects another. Examples include the production of antibiotics or bacteriocins that inhibit or kill susceptible microorganisms. Competition arises when different microorganisms within a community try to acquire the same resource. This principle, known as the competitive exclusion principle, can lead to the elimination of one species from a given habitat or resource. Microorganisms interact in the cycling of nutrients such as carbon, sulfur, nitrogen, phosphorus, iron, and manganese. This biogeochemical cycling involves the transformation and recycling of nutrients through oxidation-reduction reactions. Understanding microbial interactions is essential for analyzing human-host microbiota, developing synthetic microbiomes, and various biotechnological applications. Examples include biofuel production, bioremediation, and biodegradation of pollutants, showcasing the importance of microbial ecology in research.